My name is Mel Masuda from Scale Biosciences. We use combinatorial indexing for single cell sequencing. And so I'm going to take you through our base technology and then our RNA sequencing kit, as well as some additional applications that are launching later this year. So as we look at single cell sequencing over time, you can see on the x-axis here, the years, and then the number of cells in studies on the y-axis. And so it's a pretty clear trend that this is going up, starting from single cell per well homebrew methods at the very beginning, some of the early commercialized methods like SmartSeq, Fluidigm, and then going into the droplet-based methods, which was another step going up with the homebrew in drop and dropseq, and then when 10x launched, and this just keeps going up and up. And so we see this is something that we want to enable with our technology, and there are several applications that we think of as these studies are needing to be powered by more cells and more samples. So, classic atlasing, going really in depth on different tissues and model systems, longitudinal studies where you want to look at something over time and potentially even batch those together to get rid of batch effects. Rare cell profiling. So this is like the needle in a haystack. If you've got something that's 1% of cells, but you also want to look at the rest of the cells along with that when you're profiling. And then of course, large patient cohorts, CRISPR screening, which is getting bigger and bigger, going on to genome-wide, and then biomarker and drug discovery. I'm sure you guys have other examples as well, but these are some of the applications that we think about. So to enable going higher with number of cells and number of samples, this needs to be tackled from a couple different perspectives. One is just, the pure technology perspective. So being able to process more at one time. So we do that through combinatorial indexing, which I will definitely show you in this presentation, but we also think about it from the cost perspective. So being able to fit these within the budgets that you have and making the most out of your money. So we really tackle this problem from those two sides and think about that. A little bit of background on the company. These are our scientific co-founders. You might recognize some of these faces. So on the right-hand side, we have Jay Shendure and Cole Trapnell from University of Washington. They have co-developed all of the SCI method. So that's single cell combinatorial indexing and all of the flavors of that. Gary Nolan at Stanford, well known for proteomics work. His lab developed quantum barcoding actually about 12 or 13 years ago now. That was actually purchased by Roche. Roche didn't really do much with it. And then we got the exclusive licensing from that, but it's kind of a really early split pool method. And again, being Gary focused on protein. And then Frank Steamers as well, who's our CSO and previously led advanced research at Illumina. So Frank collaborated really heavily with Jay and Cole and was a big voice in getting combinatorial indexing out to the rest of the world and out of Jay and Cole's labs. So what we have today is a couple of kits on market. I'm actually not going to talk about single cell ATAC-seq, but would be happy to follow up with anybody who is interested in that for sure. We'll go into depth with our single cell RNA sequencing kit, which launched earlier this year. And then coming out actually quite soon, we have single cell methylation and then an extension of our single cell RNA kit to enable CRISPR screening. So I'll show a little bit of data for those two, but again, if you guys want more information, we're happy to follow up afterwards with a meeting specific to what you guys are up to. Okay, so the basic gist of combinatorial indexing is that we use the cell as a compartment. And how this differs from kind of traditional capture systems. So you can think of this here as either a well or a droplet. But the idea with most systems is that you want to get one cell or one nuclei per, that vessel. And so you have one reaction that happens within there. You lyse your cell, do your RT and your molecular biology, and then go on to your library prep. One of the constraints of these systems is, based on throughput. You only have so many wells or droplets, and you need to have, again, that one cell per droplet. And that's because if you have two within there, you're then getting the information tagged the same from both of those cells, and that's called a doublet or a multiplet. And so you're just inherently limited by the system with other technologies. So we get around that by actually using the cell as a container, like I mentioned. So what we first do with all of our applications is we both fix and permeabilize the cell. So we permeabilize it so that your enzymes and your oligos can get in, so that molecular biology can happen within it. But we also fix it. One, it makes it much more stable, but two, also so your RNA doesn't float out of it. And that allows us to go through multiple processing steps within that cell or nuclei. So this configuration is an example based on our RNA kit. But you can see in the first step here, it's a 96 well plate. You're loading many cells per well, and each well has a different barcode in there. And so at this step, you can actually use this first barcode for sample multiplexing if you would like as well. You just note which wells you put each of the samples into, and you can split that pie however you would like to. From there, you take all of your cells, you pool them together, and then you randomly distribute them across the next plate, which in this case is 384. You add on your second barcode, and then you do another pool and split across 96 well plate to add on your third barcode. So we look at the number of unique barcodes or barcode space. Why that is important is, again, that doublet rate. So for us, a doublet is a cell following the exact same path and getting all three of the same barcodes. And we want to target a really low doublet rate. It's just simple math for the output, where we usually target about 3% to keep it as low as possible. All right, so from a high level, this is our single cell RNA sequencing kit. All of our workflows are totally instrument-free, so they're plate-based based on this combinatorial indexing. I also mentioned fixation. Not only does that allow us to do the combinatorial indexing, but it also allows you to store away your samples to be processed at a later date. So if you're getting samples from different projects or customers, those can be batched together, or in the case of longitudinal studies, if you'd like to do treatments over time or something like that, you can put them all in one experiment to get rid of any technical error or batch effects that there might be in other systems. We've also done a lot of workflow efficiency and optimization in our development lab. I don't know if any of you are super familiar with like the SCI methods or have even tried them out, but they're not terribly user-friendly. So a big part of bringing these technologies to market was making them super robust and easy to use. And then we have a full bioinformatics pipeline. This is true for all of our products, but it'll feel pretty similar. I'll have a little bit more detail later, but it'll feel pretty similar to the standard processing for an RNA-seq library. All right, so for the input of our RNA kit, we recommend anywhere from 5 to 10,000 cells or nuclei. It works with both per well. You take that through the process, and for this kind of iteration of the kit, that's 125,000 cells out. And again, that's based on targeting about that 3% doublet rate. I'll note quickly that we have the ability to extend the throughput of this while still using, my mouse doesn't work. While still using the same configuration by adding additional of these final indexing plates, that split 3. So if you add another one, that would increase, another 125,000 so that you get 150,000 out of it. So if you want to profile more from that single workflow, you can, and it's pretty flexible because you can buy those plates separately, so it's totally modular. But you can also store the cells before you decide to do that. So if you wanted to go through the process, sequence this once, and then you're like, that was a really good experiment, we'd like to go more in depth, you can pull those from the freezer and then put it on the plate. And so it just makes it really tunable to how you want to do your experiment and how you want to scale it up. And then just to mention as well, again, we put a lot of workflow optimization in here, and this is all 2 days, totally instrument free. Single cell labs should have everything on hand, just some standard plastics and reagents that's on our shopping list that's not in the kit. Just a little bit more detail, and I know this is tiny, I'm so happy to send this to you guys as well. But this is just the configuration of the barcodes that are added. So we do 3' RT, so that's based on poly A capture. So our RT oligo has the poly T on there. You have your barcode, which is that well-specific barcode on those 96 wells, and then you have a UMI so that you can do your transcript counting as well. The second barcode is added on by ligation on that same side there. And then that third split in barcode is tagmentation on the other side. And then it's a simple PCR, which adds on that third barcode in your Illumina adapters as well, so that you're ready to go on with your sequencing library. So this is a seemingly, it looks very simple, but we put a lot of work into engineering this pooling funnel. And so what it does is allows you to, instead of pooling your wells after you do each reaction by, multi-channel pipette, what you can do is just take that, flip your 96 or 384 well plate in there, and then give it a gentle spin down in the centrifuge. And you can see it's kind of got like an Eppendorf tube size compartment at the bottom, and then you have one pool to go into your next molecular biology step. So we have a fully packaged workflow for our bioinformatics analysis. This is all packaged in Nextflow. It's all based on open source tools. It's open, so you can change things if you would like to. Again, this will feel really similar to other workflows out there. The big difference for us is that we have, 3 barcodes compared to some other technologies. So we have more demultiplexing steps. And because of that, we have some additional outputs just for the QC. We like to look at the performance of each of those barcodes at each stage. So you can see, maybe there was something sample dependent and the RT from the wells of a given sample didn't work quite as well, just to allow you to trace back and look at the steps within the process. All right, so a little bit of data. Classic barnyard experiment. So what you do here is mix together 2 species, human and mouse. You put them together before going into the workflow. And what that allows you to do is assess your doublet rate and your background rate. And so that's exactly what we did, mix them 1-to-1, took them through. One thing I like to point out here is it says 10 wells taken through sequencing at the end. What you can do from this last plate, there's enough reagents to actually create 2 separate libraries. So we do this all the time in R&D, where we don't want to, maybe we want to just sequence, a row of cells, and you get the full depth from each of those cells on like a NextSeq before you go on to your NovaSeq or whatever sequencer, just to get an idea of the data quality. So again, it just gives you some flexibility because these are big data sets for how you analyze and process everything. I mentioned 3% doublet rate a couple times, right around there with this, low background at 2.8%. We consistently see really low background. And one of the cool features of our kit is because we aren't, one well or one droplet where everything happens, we have built in kind of wash steps throughout the process. And so any ambient RNA or anything like that just inherently goes away with our system without a lot of extra designer work there. And then just to point out too, so these are cell lines expressed really, really well, but at 60,000 reads per cell, we're at about 15,000 UMIs. All right, a little bit more interesting samples coming up. So this was actually a customer PBMC sample, and they wanted to fully profile this across one plate. So they wanted the 125,000 cells from the PBMCs. Right-hand side, you guys are probably used to looking at knee plots, but the big takeaway here is you want a really good signal, which is the top portion, to noise on the bottom. And you also want that knee to be really straight. If it's more angled, that's indicative of having a higher background rate. And so we're really happy with this data, clear distinction between signal and noise. And then we took this onto sequencing. I mentioned 125,000 cells. Customers often get a bit more than that. So that's kind of the floor of the kit, as long as your samples are pretty good quality. So in this case, they got 161,000 cells out. And this was sequenced to about 22,000 reads per cell, with about 2,000 genes and 4,000 UMIs coming out. And so they were actually looking at comparison with the same sample to other technologies that are out there and, on next versions of kits. And we performed really well with this same sample. So we were excited to see for our V1 kit getting really good data quality here. How am I doing on time? Okay. So of course, the next step after looking at those really high level QCs is that you want to do your unbiased clustering and check all your markers. So You can see the UMAP on the left. The high level check here is just to make sure that all of the appropriate cell types are represented and are within the data set. And then on the right-hand side, looking at specific cell types and making sure that marker genes that, should be highly expressed are there. And then, marker genes that shouldn't be in the same cell are not. And so this all checked out with this. If you ask me too much about PBMCs and immunology, I will crack quickly, but the customer is happy with this data. Another thing we wanted to do was compare our kit after, all the development work that we put in to the skyRNA, what is now V4 protocol. So I think it's been about 10 years that Jay and Cole have been working on Sky methods and improving it. And they put out the V4 last year. So this was run in Cole's lab. He's really big into zebrafish development work right now. So this was 4 different zebrafish nuclei from those zebrafish through one workflow. Again, they got 170,000 nuclei out, so higher than 125. And then the high level takeaway here is that on the same samples, they got about 50% both more genes and more UMIs than they did with the homebrew method. So that was really great validation for us that all that work that we put in really paid off. And they were very complimentive, especially of that funnel, which they use pretty regularly now. And this is a pretty common way to compare to the technology that you're currently using, because people want to look to see, can I switch technologies, get similar data, compare to my old technology, so that switching mid-project is actually feasible. So on the left-hand side is the UMAP, the overlaid of both of the Sky method and the Scale Bio kit. And on the right-hand side, they're just split out so that you can see them a little bit more distinctly. As you can see, the clustering is super similar. All the same cell types were observed that were expected. And so for them, this means that they can, with this project, move forward with our kit versus the homebrew method. And this is from our beta tester at UCLA. Dan Geschwind actually gave a webinar for us on Tuesday. So there's a lot more data. They've done tons more work with our kits on a couple different projects as well. So we'd be happy to send that to you guys if you want to hear him talk about it. But end of last year, they really put the kit through the paces and did a couple different things. So they mixed both cells, primary cells and nuclei on the same plate. And then they also used nuclei from macaque brain that had been stored for like 7 years in the freezer. So they really tested us out here. The output, they got 188,000 cells and nuclei mixed, and they'd previously been using a lot of 10x for this, this project. This person was specifically interested in the macaque nuclei, so that's what the analysis is here. So again, the UMAP on the right is the overlay of the 2 technologies where we match up really, really cleanly with their previous data. And actually, they were able to see a couple additional cell types that they didn't get with 10x. And then you can see in the middle here, they actually got 50% higher gene sensitivity with our kit than they did with 10x. And I think kind of anecdotally, or a guess as to why that might be, is the Sky methods were developed on nuclei, and we actually did the work to make them work on cells as well. Jay and Cole do pretty much exclusively nuclei. And so we perform really, really well with difficult tissues compared to other technologies. So this is just to show some of the ways you could imagine setting up an experiment. Again, each of those different wells at the first step have a different barcode, so you can split this however you would like. A few examples here, pretty common is people do about 12 samples per plate, and that means you get about 10,000 cells out at the end. Basically, what you do is you get about 1,300 cells per well out at the end. So you just do the back math to figure out how many wells you load per sample at the very beginning. You don't have to split them evenly either. You can do half a plate that's 1 sample and then 5 other on the other half. It's really choose your own adventure here. All right. So again, high level mentioned the fixation where you can then store the samples, and that's up to 3 months right now. We're doing additional validation. Those are just still in the freezer. So expect that to be pushed out as time goes on, and we just test those samples. 3 levels of indexing, totally instrument free, flexible design, and then we fully support through the data analysis as well. So maybe I'll just take a quick stop there and see if there are any questions on the RNA front. Yeah. Yeah, so just for online, the question was, will the fixation decrease the RNA content? Is that right? Quality, yep, okay. So what we've seen from a few of these samples is that we actually get similar, if not better, data quality to non-fixed technology out there. So again, the PBMCs were similar to 10x and other technologies out there. And then with the nuclei, we actually got more. So we don't see any limitation with the fixation actually. And it's a DSP-based fixation. Correct. Yep, you don't take a hit there. Yeah. Oh, the workflow? The last one I was on? Or? That one? Sure, like the barcode, the different molecular biology steps, or how the barcodes are added, or? Yeah, so again, we do, so 3 levels of indexing, or we're adding 3 different indices on each of these plates. So in that first 96 well plate, you're doing RT, and that's based off poly A. So we have the poly T to prime to get your cDNA. Then you pool and you go on to the next plate, and that's when we do ligation. So you can see on the, it's even very small for me. The ligation barcode is the red one here though, and that's your index number 2. I don't have an exact answer for the ligation efficiency. That's something I'd have to check with internally with R&D.
Well, must be pretty good.
Yeah. Yep. Yep. Yeah, and then the third step is again that pool and then split, and then we tagment on the other side, and that's what gives you the appropriate library length for Illumina sequencing as well. And then you add on your last barcode and your Illumina adapters via PCR. Does that make sense? Okay, great. Oh, sorry, yeah.
I was surprised to hear you say that there's no any tested that.
Well, the fix and perm happens at the same time. So I think everything just kind of gets locked in there. And then again, throughout the process, anything that is ambient or like dead cells would be washed away. So I don't know if we've done any experiments to kind of just test if anything gets out, but yeah, based on the RNA quality, it's sticking in there pretty good. But that's another thing I could check on just for curiosity. Yeah, either one. Yep. Correct. Yep. So you want to use all of your barcodes. Otherwise, if you used a half a plate at the beginning, your doublet or multiplet rate would go up a lot higher. So you want to use every barcode in every experiment. Yeah. I'll answer and then see if I actually answered the right question. But so you can do that however you like. You just need to pay attention to which wells you put your sample in. But I'm not sure I fully understood your question.
Like say,
Uh-huh.
there could be like different some of the quoting.
Mhm.
So I need, like, say, the person picks out, well, to be, like, one more with the thing. Or should I make all of them?
Oh, yeah, you'll keep your samples separate and distinct and then load them onto the wells. So like if you, maybe this is back to here, sorry. Oops. Oh, wow. I clicked too fast. Now it's mad. Okay. So yeah, if you pretend this middle one is organs instead of people, you would just like if you wanted about 10,000 per, you would load one column in this case. But keep them distinct before going in. You'll fix them separately and then put them onto the RNA plate, and that's when you would mix them after that. Correct. Yep, everything gets mixed together and then randomly put across the next plate. Yep.
You, like, based on your nonprofit sector, does it mean that the cell all of them matter? Because like in ag, they not do like cell that large, irregular shape.
That's a really good question and something that a few of our customers are testing right now. So like cardiac cells, adipocytes, stuff like that. We haven't pushed the limits in our R&D, but certainly something that people are excited about. So stay tuned. We'll keep updated with how that progresses. And I think there are some questions here. Let me just really quickly. Has the technology been used with a wide range of tissues? Yeah, so lots of customers have done brain, liver, kidney, I'm blanking right now, plant tissue, all kinds of cell lines, primary cells, even some more difficult like OCT embedded stuff. Yeah, so it's been put through quite a few different things beyond, model organisms at this point. What's the reason for limiting the plate to 12 samples? In this case, it's just if you're desiring about 10,000 cells per sample, you could load 12 samples. On the right-hand side, you can see an example of 48 samples where you would get about 2,600 per. So it's not limited at all. Again, you get about 1,300 out per well. So then just multiply that by however many cells you want per each of your samples. Human brain organoids, I don't think we have an example of that right now. Yeah, this is a good question to point out. What is the recommended number of cells or nuclei going into fixation? We have two protocols. So we have a large volume protocol, which is 400,000 cells or nuclei at the start. And we have a low volume fixation protocol, which is we recommend about 50 to 100,000 cells or nuclei going into it. We still get output if you do less than that. It's just really hard to count them accurately. And so that's kind of the lower limit we're comfortable recommending, but customers certainly do go lower to that and they just get out what they can from it and they still get data from it. It's just a little bit harder to control at that point.
So your capture rate is 10% below volume. So I'm saying you start with 100,000 per 10 to get 10,000 cells again.
So these are, I think, two different things. The fixation capture rate with the high volume, we get like with good quality cells, there's always the asterisk with single cell work, as you know, but like 80 or 90% back often. The low volume, we still get like 60 to 80% back. It's just really, really hard to count when you have very few nuclei or cells. For our kit, the capture rate is the total input is between 500,000 and 1 million cells or nuclei. And then the 125,000 output. But like I mentioned before, we can get more out of it by adding more of those final plates. So it's kind of a moving target that's a little bit hard to answer, but our capture rate can be as high as like 50% with that. So it just depends on how many you want to sequence at the end. All right, I'm going to move on for the sake. Oh yeah. All right, speed round here. So CRISPR screening, obviously an application that takes a lot of cells, especially as we're seeing more and more people wanting to do genome-wide screens and have multiple targets or multiple guides per target. So I'm going to skip kind of the background of the CRISPR screening. and go just into the differences of the RNA workflow for our kit. So this is based on the backbone of our RNA workflow. So everything that was true for that for RNA quality is going to be true for this kit as well. We've just added some additional stuff here to make it, to increase the guide capture. And so with CRISPR, you really want your guide to know what you've targeted or what you've perturbed to match to the RNA data. You need both of those to get, an actual solid readout from this. So that guide capture is a really important metric for single cell sequencing from CRISPR screens. So we are compatible with crop or crop-like libraries. You obviously do your pooled screen ahead of time, fix your cells, go into the RNA workflow. I think I'm gonna skip ahead really quickly. One of the big differences here, we've increased guide capture kind of at 2 steps. The first step is in that split 1 or the RT barcode, where along with that poly T primer for poly A from 3', we also have a guide specific primer in there. And so you're getting RT amplification of your crop guide. Ligation is the same, indexing is the same, and then we take a small portion of the library to do a guide specific PCR. Again, it's like 0.25 nanograms, just a tiny bit of that. And so all of that is meant to boost up your capture of the guide library. Let me just go back and see if there's anything else here. So again, all the same stuff with the RNA workflow, really high throughput, cost per cell is much lower. Again, you can fix these and store these. And then we have a CRISPR specific pipeline for this, which does your guide detection as well with your RNA and then matches them together for you. And then again, this will be modular too, and flexible. So if you sequence those 125,000 cells at first, you can take those cells out of the freezer and then purchase the kits, the final distribution plate kits separately to profile more cells later if you would like to, or you can do it all at one time. It's totally up to you. I'm going to skip some slides in the interest of time here. Sorry. So again, a big metric for CRISPR screening is that guide capture. So we wanted to look at the difference between poly A only, because CropSeq guides do have a poly A, or CropSeq constructs do have a poly A on there. But we also wanted to compare that to having that custom guide primer or that CRISPR construct specific primer in there. And what you can see on the right-hand side is how we measure guide capture. So the threshold here is 3 or more of that guides read per cell. You can change that threshold in our bioinformatics to, if you want it to be more stringent, you can. If you want it to be less stringent, you can do that as well. The key takeaway here is that on the right-hand side, you can see with that custom guide primer, we have more cells with more guides in them. And so your guide detection is much more sensitive. The blue line just shows the percentage of guides that are captured within the system. So with both of these, you can see you're getting on the left-hand side, 100% of your guides or 100% of your, perturbed targets, but that falls off really quickly. And if you're perturbing things, you want the readout from that. So you want to assess your entire guide library. Does that make sense for that? Okay, cool. And then we've tested this in a few different systems. So of course we optimize on cell lines in-house. We get great capture with that 95%. With some early customers, we've also done some much more difficult samples where these cells don't express RNA as much as cell lines do. And so you would expect to take a hit there. And we're still at 70% of guide capture with this. So we're pretty excited about this system. All right, that was the real speed version of CRISPR. Anything to go over there? Okay. And then we're super excited about single cell methylation as well, because this is kind of a differentiator for us. There are a few different things that people look at from a single cell level. So, classically a ATAC-seq RNA protein, but kind of one step ahead of that is single cell methylation, where an area that's demethylated can then be made open to have the open chromatin or ATAC to then be made into RNA and then protein. And so this is giving you kind of that earliest signal and often the most stable signal as well. So this is, again, totally plate-based, based on combinatorial indexing, has fixation in there. It detects both CG and CH methylation sites. And then again, we have a bioinformatics pipeline to go with this as well. So this workflow looks a little bit different. This is 2 levels of indexing. And what we do is, this is all nuclei going in because you're looking at the chromatin. So you have your single nuclei suspension, you fix that and then do a nucleosome depletion because we want to assess the whole genome and not just the open chromatin. From there, you do tagmentation, and this is your first index. So that goes and randomly distributes tags across the genome. And then you pool those together and then go onto your next 96-well plate. Right now, we do have flow sorting in the process. It's just to get a really consistent number of nuclei per well. But we have shown and actually have some data that it looks quite good, even if you do that just by pipetting or dilution, like it evens out enough that it works just fine. So after you go onto your second plate, that's when you do bisulfite conversion. And then you add on the second combinatorial index through ligation and PCR on your library, and you're ready to send it to Kristen for sequencing. Oops. So we've tested this internally and then with some early customers across quite a few samples at this point, again, starting with some of the model human and mouse systems, PBMCs as well, and different tissue, including frozen tissue. And I have some data from OCT embedded frozen tissue. So really, putting it through some of the more difficult sample types out there. So this shows, this is really small and I apologize for that, but this shows a couple of different metrics that people think about for kind of high level QC from methylation. So you think about the number of unique reads, so unique reads from the genome that you get. So that's this first and third run. And then also people think about CG coverage, so that the coverage of the methylation that your sites that you're getting over the genome. And so I think the most interesting one here is probably the one on the right-hand side, which has all of those samples that I listed before. We were really happy to see that we get super consistent. It's total reads on the x-axis and then unique reads on the y-axis. It's linear, we're nowhere near saturation here, and we get really similar and robust performance across various sample types. So this is just a quick test to show that you can do this without the flow sorter if that's a limitation or you'd rather not have that within your workflow. You can see that the knee plot is still quite steep there, which is good, but the background is a little bit higher on that right-hand side, which is the unsorted side. You don't take any kind of hit to the percent of methylated GC, so the data quality for the cells that you get is good. It's just going to have a little bit higher sequencing background. We're still optimizing this in-house too, I will say, but we just wanted to look at this to show that this is possible and to make it completely instrument-free. And I know I'm rushing through this, but I know we're almost at time too. So quickly wanted to show that most kind of extreme example of the frozen OCT kidney scroll. And so this was a pilot test for a customer where they had from mice, 3 different mice, kidney nuclei that they wanted to profile. So you can see kidney A, B, and C. I'll just note that kidney C is scooted to the left just because they had less nuclei. It wasn't a very good prep going into it. So it's just less cells, but you can see that the signal to noise is still quite good with that one. They wanted to profile these really, really deeply. So they wanted, above 10,000 nuclei for this first test, which we got. We looked at all the sequencing metrics and we're really happy with the passing reads and nuclei. And then one thing to note with single cell methylation sequencing is that because you're looking at essentially the whole genome, it's a whole different game with sequencing depth. So instead of being, the 20 or 50,000 that you might do with RNA, people are more in like the 500,000 to 3 million reads per cell. So this customer, again, wanted to go really deep and get all the information that they could. So this ended up being 2 NovaSeq S4 runs, which is about a little over 2 million reads per cell. And that got 1 million unique reads. And the big metric that they wanted to look at was the CG sites covered, and they wanted above 500,000, which we got here. So they were super excited with this. We were, again, excited to see this work on such a difficult sample type. And then I'll just mention this really quickly. This is not just for methylation, but we do have services available. So specifically, people seem to be interested for methylation just because it's a newer technology and they'd like a data set to get their hands on. So what we can do is basically enable you with the fixation portion. After we design the experiment together, that can be sent into our development lab up in San Carlos, where we will do the library preparation. We can do sequencing, or sequencing can be done elsewhere. And then we can do the analysis for you if you like as well, or provision you with the pipeline. So this is an option for people who want to get to some data to play around with quickly. Again, any of our workflows here. All right, so that's what we've got. Questions on methylation or anything overall? It's over the whole genome, but it doesn't cover the whole genome, if that makes sense. Oh, sorry. So the question was if TN5 insertion is limited on the genome. And so it's randomly distributed across the whole genome. You're not going to get whole genome coverage of each single cell though, but when you get enough cells in a cluster, you can put those together and get whole genome coverage for a cell type, if that makes sense. It depends on what the vector is. We need some length for that tag mutation enzyme to get into. So if your guide is at the very end, this version of our kit won't work with it. I'd be happy to look at your guide construct if you would like, and chat through that for sure. And then just quick questions online. Do we have any specific dissociation protocols for tissues? We would usually leave that up to you guys. You're usually the experts in your tissues, not us. But we could point you to some customers who have had success if it's similar tissues. And then what's the lowest cell viability recommended for single cell prep? How would you recommend to process low viability samples? You definitely want to have as high viability as possible. I think that's true for every single cell workflow out there. So yeah, dead cell removal can be good if you have enough going in. But kind of the quality that you put in is the quality that you're gonna get out there. And then is your RNA kit compatible with ATAC as they're currently using Multiome? The answer is no. We do have the ATAC kit that's separate. Again, I didn't talk about that today. You guys can reach out and we can talk through that if you like. We are, after we get all of these single modalities down, our goal is definitely to mix and match all of these and get as much information as we can from each individual cell. Yes.
So if you have very different cell types
Mm-hmm.
across the plate.
Yep.
Let's say somebody wants to use hepatocytes, somebody wants to use lymphocytes.
Yep.
Do you see that the recovery is not cell type dependent?
Mm-hmm. So Kristen is asking about having pretty different cell types in the RNA plate and the considerations for that, and if the cell capture is similar. So we do see similar capture across them. The one asterisk I would put on there is you don't want things with really, really disparate RNA content. So I wouldn't put like those zebrafish embryos with 500 UMIs in with K562s or something like that, because you're just going to have to sequence through the library of the higher expressing cells so deeply to get to the information for the really low expressing cells. If it's pretty close, it's totally fine. It's just those ones that are really, really far apart where you have to spend more on sequencing. So. All right. Thank you all.